Assalamu alaikum wa rahmatullahi wa barakatuh. Good day, everyone. Welcome, talented students, to lecture number 3. In the previous sessions, we explored the basic idea of the cell as fundamental unit of life. Today, we will go inside the cell to understand the tiny structures, organelles that work together, like perfectly organized factory. And all that in biology phase 1 from NSMO program. This is your instructor, Muhammad Al Ali. I salute you. And we still in chapter 1, cell structure and function. In the previous lectures, we talked about the cell, characteristics of living organisms, cell theory, differences between plant and animal cells, and we talk some of organella, which is ribosome. And today we will continue discover the components of the cell. Welcome everyone to lecture 3 in our biology phase 1 from NSMO program. Today we are diving into the fascinating world of cell components, the tiny structures that keep every living organisms alive and functioning. Before we begin, let's briefly recall two key ideas we covered in the previous lecture. The cell theory, which explains that the cell is the basic unit of life, and the types of cells, prokaryotic and eukaryotic. These concepts will help us understand today's deeper discussion. In this lecture, we will focus on two major areas: the components of the cell, the internal parts that perform essential function, and the cellular structure and its parts, how these components work together as a coordinate system. Expect clear explanation and visual learning throughout the lecture. Well, let's begin with the endoplasmic reticulum, a network of interconnected channels. There are two types of it. The first one, rough endoplasmic reticulum, and we called it RER. That one, guys. RER. So it's covered with ribosomes. Look, this is the tiny things. We called it ribosomes, making it's the site of a protein synthesis, because ribosomes is synthesis protein. So basically, if it has ribosome, it will also synthesis protein. The other one, smooth endoplasmic reticulum, SER, that one, SER, lacks ribosomes, and it's responsible for lipid and carbohydrate synthesis. So, liver cells, the SER plays a detoxification role. So this is main things in liver. Okay. The second organelle, we'll talk about it, Golgi apparatus. See it in these figures. And it's a system composed of Stax membrane. Okay. Modified sorts and packages materials. It's like packages some materials and packages materials in sacks called vesicles. This is called vesicles. And these vesicles, after that, it will be formation to lysosome. Okay? And lysosome, as we said before, it's only existed where, guys, do you remember? It's only in animal cell. Don't forget about that. Let's talk about another organella, lysosome. They call it digestive bodies. Look to this diagram here, the lysosomes. It shows the process of engulfing particles and breaking them down. So, lysosomes sometimes act cleaning crow. So, they digest like this damaging mitochondrion or unwanted food from food vessels. So, also, the lysosomes be part from what we call the program cell death apoptosis. Apoptosis. Okay. Let's talk about another organelles, vacuola and vesicles. So they are responsible for storing materials. Okay. And we see it in a planet, it's too large because the process photosynthesis that produce food, and it's very, very small in animal cells. And in animal cells, it's smaller and probably it will be multiple vacuoles. Next, let's look to another important component, the centrosome. And the centrosome, which contains a pair of structures called centrioles. That's the first centrioles, and it will be the another one here. So, as you see, the image, each centrioles is made of cylindrical, that we call this cylindrical, that one, microtubules, and it's very organized patterns. And these centrioles are responsible for forming the spindle fibers during cell divisions. So this makes the centrosome essential for animal cell divisions. Animal, it's not existed in a plant because spindle fibers help separate chromosomes accurately. So without centrioles, the cell would not be able to divide it properly or distribute genetic materials correctly. Don't forget. Now that we have learned about several important cell organelles, such as endoplasmic reticulum, Golgi apparatus, lysosome, and vacuoles, let's apply this knowledge with a quick exercise. So, the questions asked, the taste of a planet differs from one another because of variations in the contents of the what, guys? Is it vacuolus or cell wall or plastids or lysosomes? Okay, so the answer is, what is it? What is it? What is it's vacuolus, because the vacuoles store many substances inside plant cells, including sugar, organic acids, salts, pigments. So these stored chemically directly influence the plant's flavor, smell, and even color. So when the contents of vacuoles vary from one plant to another, their tastes naturally change too. Cell wall, it's about protection. Plastid, it's about photosynthesis. Lysosome, it's about digestive or analysis unwanted organelles out wear or waste. Okay. Today we covered the major cells components and how they function integrated system, and we understanding these structures is essential for deeper study in biology. Thank you for your attention. I'm proud of your engagement and curiosity. See you in the next lecture. So long.